All right, hi everyone. Welcome to today's micro seminar. It is my absolute pleasure to introduce Dr. Jennifer Glass. Jen is currently an assistant professor at Georgia Tech, but is up on the promotion scale this year. So good thoughts for her. Jen started her career at ASU upstate from, I guess, depending on how I'm thinking today, it's the second or third best city in Arizona and Phoenix. After her PhD at ASU, she did a postdoc with Victoria Orphan at Caltech, and then started her lab at Georgia Tech in, I'm going to say 2013. It might be 2014. I think it's 2013 though. The only other thing I want to say about Jen is she has been, she's a great scientist, but also a very positive influence. At least I can say this for me. Kyle's going to say hi. I can say this for me because she's been a very positive influence online for both inclusivity and diversity throughout sciences. And so I can say that it's impacted the way I think about things. And so I just want to say thank you for that. But other than that, take it away, Jen.
Thanks so much, Dave. That's a really nice introduction, and I really appreciate all those comments. I've also learned a lot from you on Twitter, and in fact, I think that micro seminar is just such a beautiful example of how social media can be used to create such new spaces and such more inclusive spaces for science. And I really do believe that this is the future of science. We're just using electrons. We're not using any carbon right now, and that's fantastic. And so I just want to thank everyone who brought this community together, and I'm really honored to be presenting. And I know this is being recorded, but I guess I'm a risk taker. So instead of presenting to you things that I know I can talk about, I've given seminars on previously, I'm going to talk about new stuff, really new stuff that's challenging for me. So I'm excited. I'm also a little bit scared, but that's good because we're all students and we should all always be learning. So I'm trying to put myself out here to do that as well. So I hope I can ask your questions, answer your questions, ask questions too. If I can't, I'll just say I can't, like in a comp exam, and we can talk about the answers in the future, because a lot of this I think is still, my group is still sorting out, and I'd love your ideas and feedback. So, all right, without further ado, I'm going to be talking today about a subject that's become very near and dear to my group at Georgia Tech, which is deep breathing. And by deep, I mean deep in terms of time, but also deep in terms of space, because a lot of us here, of course, love the deep biosphere and love all these interesting genomes that are coming out of the deep biosphere and that continue, at least for me, to cause me endless challenges, but also endless fascination. And I really do believe that the genomic information coming from the deep biosphere is really going to fuel our field for many years to come. And I really hope that increasingly we can also turn to biochemistry to start to get at characterizing some of these proteins. So that's something we're hoping to move towards as well in the future. All right. So I'm also going to talk about deep breathing a bit through time as well as space. So first of all, I'm going to just give an outline. So there's going to be kind of two acts to this talk, both of them with this deep breathing element. Of course, breathing being respiration. First, I'm going to give a review of what respiration is. I'm going to talk, I said I would talk just about anaerobic, but in fact, anaerobic respiration is really thought to be where aerobic respiration came from, and I'll talk about that. So it's kind of important, I think, to set up that transition. So I'm going to talk about both, or I'll try to talk about both. And then I'm going to move on to what we're seeing in some of our genomic bins from 2 different deep biosphere systems, one deeper than the other. One is a ferruginous lake metagenomic bin that my postdoc Nadia Seinbaum is working on, and the other is from a methane hydrate sediment from Hydrate Ridge off of Oregon, and that is genuinely deep. That's 70 meters. So, okay, and I'm going to talk about what we're seeing in those in terms of, I'm not going to talk about the full genomes, I'm just going to talk about one per genome, potential new respiratory complex. You can evaluate whether you believe it could really be respiratory or something else. So first of all, something that we don't do enough of is just, or I don't do enough of always, is just to stop and breathe for a second. So we all can take a big breath in and out and just enjoy that. Appreciate the fact that we have this 20% of our atmosphere is molecular oxygen, and that is what is enabling us to breathe right now. That's really the only thing that we humans can respire. So that is aerobic respiration, right? Aerobic respiration is defined as molecular oxygen being reduced to water with electrons coming from this electron transport chain. And in the process, very importantly, protons are being translocated across the membrane, and that proton motive force is being used to drive ATP synthesis. So here, I'm going to try to get a bit more into the biochemistry. And because I want you to be able to go back and check everything I'm saying and go through it slowly yourself, as I like to do with biochemistry, because it can be very complicated, I'm giving you all the references at the bottom of the slides, but I'm also going to give you the full references at the end of the talk, which I think will be very helpful if you'd like to read some of these fantastic papers on these subjects that go into much more depth than I'll cover here. So this is what we're all doing right now, aerobic respiration. And this is the respiratory electron transport chain. And I'm mostly going to talk about the evolution of 2 complexes, complex 1 and complex 4 today. So the first one and the last one. But of course, very important things happen in the middle too. And another day we can talk about the evolution of that. So in a nutshell here in complex 1, we have NADH being oxidized, and we have quinone being reduced. This is also known as NADH, ubiquinone oxidoreductase, and it's a huge machine. It has different modules. There's this module here, which is known as the N module, I believe for NADH oxidation, N module. And then there's this kind of transporter-like module. I'll talk more about this later. And this is actually where the protons are being translocated across the membrane. So it's very important. And it turns out it actually evolved from a sodium proton antiporter. and kind of merging then with different kind of hydrogenase subunits. So a kind of common theme throughout this talk, and just kind of looking very quickly, glancing at this figure, you can see that there's multiple subunits for every respiratory complex here. And as maybe isn't surprising, but I think is quite interesting, is that these are can kind of be thought of as building blocks that came together in different organisms in slightly different ways to form these complexes. So I kind of went over quickly what complex one is. That's the largest one here. And then at the end, the terminal oxidase is complex 4, which is more commonly referred to as cytochrome C oxidase. And so the site at which the oxygen is actually being reduced to water as we breathe oxygen is this copper site. And it's talk about later in the talk about how we think that this copper site came to be. And you can also see that there's, very importantly, these proton translocation pathways again in this enzyme. So each of them has a name. There's the D pathway is very important for proton translocation. Also, there can be other pathways, the K pathway. So that is, in a nutshell, aerobic respiration. Of course, you learn much more about it in your classes, but I just want to give a brief overview. And reminder that 10 protons are being pumped per electron pair in this process. And that, to my knowledge, is actually the most protons pumped in any known respiratory complex. So as far as I can tell, this is really why aerobic respiration has, in a sense, kind of organisms that do aerobic respiration have kind of taken over the surface world, is because it, for various reasons, not just thermodynamic actually, but more on the kinetic side and on the very kind of optimization and efficiency of this process, which is beautifully described in this paper by Chen and Strauss, 2013, that I really encourage you to read. This is really optimized for aerobic respiration in these complexes, is really optimized for proton pumping. And it's actually been a few recent grant reviews that have forced me to think more about proton pumping as I try to imagine different potential microbial metabolisms. So I thank the reviewers that have pointed this out to me, that I need to be considering protons and where they're coming and going from, because that has really made me think much more deeply about what is possible with different respiratory pathways. So moving on, in terms of deep time, which is really my background, I came from I still am in an earth science department, and I was trained actually in geology and ocean sciences. So all of the microbiology and the biochemistry is things that I've been learning ever since I graduated from undergraduate. So I try to think of everything, my group tries to think of everything in terms of Earth history, right? Because these enzymes obviously have been around way, 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 way longer than we have. They've been around for millions and billions of years. And Earth evolution, Earth's chemical evolution, has greatly influenced the evolution, the course of evolution of these enzymes. And that's always been something that's really been interesting to me. And it's very difficult to try to track back the exact timing of these events. But we can kind of try to piece together, as I'll talk about today, a bit more about at least about kind of the course of how this evolution happened, at least on a relative scale. So back to this slide. So it's important to consider that the atmosphere has only been really had any significant oxygen in it for half of Earth's history. And the first half of Earth's history, it was almost completely anoxic. There was really negligible oxygen in Earth's atmosphere. And that would have just, the first half of Earth history, therefore, life as we know it would have just been drastically different because it would have had to be basically solely anaerobic until the Great Oxidation Event. That doesn't mean that aerobic, that there couldn't have been some scarce oxygen. There's definitely, we think, cyanobacteria producing some whiffs of oxygen at this time. So aerobic enzymes could definitely have evolved back in the Archean, but really not probably kind of proliferated until billions of years later. All right, so what did life breathe before oxygen? Right, so, well, there was anaerobic respiration. So what is anaerobic respiration? It is use of an oxidant other than oxygen for that respiratory electron transport chain. And in the process, the proton motive force has to be established in order to generate ATP synthesis. And I'm going to talk about a bit about different types of electron transport chains because I've shown you the one that we use, and I'll tell you about other kind of modules that can plug into there, but there are many others that look a bit different, quite a bit different, that can also be used depending on the electron acceptor. So this is a really beautiful, I think, diagram from Paul Falkowski's science paper from 10 years ago now that shows basically different microbial metabolisms plotted against electrode potential. So these are more oxidized compounds and more reduced. And of course, thermodynamically, we get the most, we would predict we get the most energy conserved when we pair an electron acceptor that is the most oxidizing with an electron donor that is the most reducing, so that we have the biggest change here in potential.
Hey, Jen, I'm just gonna pop in. Do you have a little ClickUp box at the bottom of the screen? I think.
Yeah.
Hide. It says just click hide because it's.
Oh yes, I didn't know if that was
okay.
Yeah, great. Okay. Oops. Let's see. Okay. All right. Thank you, Dave, for that. All right. Good. So hopefully you can see this better now. All right. So there's volts down there. Okay. So this is what I was talking about, of course, earlier, aerobic respiration with oxygen. So that's placed here because oxygen is on the more oxidizing end of the scale, and we would predict, right, that we have a greater change here in potential, and therefore we might predict that oxygen respiration is a more favorable process, and indeed it is, and that's part of the reason why it's proliferated on the surface of the Earth. And then going down from here, we'll talk a lot more about denitrification and nitrate and nitrite respiration. But in canonical, when you learn about this kind of process in classes, this is often kind of the redox ladder that you'll go through, is with oxygen being predicted to be most thermodynamically favorable. When you run out of oxygen, you being a facultative anaerobe that can also reduce other oxidants. Then if you are able to denitrify, you would use nitrate or nitrite. Once you run out of nitrate or nitrite, if you're able to do manganese respiration on solids, that's the next most favorable, followed by iron. And then we get down to the least favorable, sulfate reduction and finally methanogenesis. And roughly, very roughly, it's kind of thought that these evolved first, that fermentation and methanogenesis and sulfate respiration were very early, and that the rest kind of evolved later, with probably oxygen respiration being the youngest. So that's very general, but I'll try to talk a bit more about, a bit more specific in a few slides. So thinking about what enzymes the organisms are using for these processes. There are a few wonderful papers I really encourage you to read. This is one of them that really beautifully goes through and categorizes the different subunits of these proteins, these respiratory complexes, by their cofactors. And they postulate that we can really think of these respiratory enzymes like a construction kit, basically, with these different domains, basically characterized by, again, the cofactor. And the cofactor can be this is the cofactor that is doing electron transport. Can be molybdopterin. That's the MO. Molybdenum is very good at 2 electron reactions. Also, iron sulfur clusters, full iron proteins like in hydrogenase, and then and there's a lot of different iron sulfur cluster proteins, and also cytochrome heme containing proteins are very important, as is obvious when we think about the term cytochrome C oxidase, right? The electrons are coming from cytochrome C. Therefore, obviously, cytochrome is a very important component for that enzyme. So this is just kind of displaying how we can put together these different modules, or nature has put together these different modules to form various different respiratory complexes. In some cases, these can just be used alone, and then in the case like in the case of aerobic respiration, we need to use 4 of them in series, complex 1 all the way through complex 4. And this paper has been very nicely followed up more recently by this paper here. Give the full citation at the end. And this is more specifically in terms talking about the evolution of sulfur metabolism. So I encourage you, especially if you're in the sulfur cycle, to take a look at this paper, because they do a really good job. I'm not going to actually touch much on this, but I encourage you to look at that if you're interested in the sulfur cycle. And this paper also really, it's the same concept though. Essentially, they're talking about these modules, this construction kit of basically different modules that are put together to form these respiratory complexes. One that we often see, and especially if you've done work on anaerobes, you see this in methanogens, in archaea, but it can also be in anaerobic bacteria, is heterodisulfide reductase. As the name implies, this is probably evolved out of the sulfur cycle. And it seems to be really important in various anaerobic respiratory pathways. And I'll come back to this actually later when I talk about the one that we think we found in our metagenome. And then you can see also here just highlighting, so these brown boxes are the molybdenum cofactor. That's this little splinter right here is symbolizing that. And the really cool thing about molybdenum, like I said, is that it's really good at these 2 electron oxidations and reductions. And it can, these enzymes have kind of been evolved to different specificity for different substrates. And there's lots and lots of different substrates that can be used for molybdopterin enzymes, from the nitrogen cycle to sulfur and many other, arsenic and many others. All right, so moving on. There's a lot of details there, obviously. So I encourage you to look into this paper for more on that. So coming back to complex 1, the first complex in our electron transport chain. I've been reading up on this because, as I'll show you in a few slides, we've been looking into something that looks like a complex 1 in one of our Ager Bacteria genomes from the deep biosphere. And I found this paper and several others really interesting. There's been a lot of nice work done on the evolution. Again, this very modular evolution of complex 1. How did these different modules come together? And are there different forms? Do we see this elsewhere in nature, kind of different varieties of complex 1? And indeed, we really do. There's a whole family of kind of complex 1 variants. So this paper talks about the last common ancestor of all of these, and then evolving off from there actually is hydrogenase 3 and 4, as well as the last common ancestor of complex 1, which went on to then form this common ancestor of complex 1, of course, eventually became, as the name implies, the classical complex 1 that we're using right now to breathe. And, but not just that, it also became the F420 dehydrogenase that's in archaea and anaerobic organisms. And also, there are kind of smaller versions of this complex 1 that are kind of in different clades. There's an 11 subunit complex 1 and a 12 subunit complex 1. But as you can see, just kind of glancing at these, they kind of all have this similar look to them with this transporter, this transmembrane component, and then this other kind of component that's kind of hanging off inside in the cytoplasm. And so I'll talk more about this when I get a few slides more. So this is I think, getting towards the end of my introduction. Of course, I always think I can go faster than I really do. But I want to highlight a paper that I think is a really creative way of looking at this respiration through deep space and time. And it used these profiles that probably look familiar to those of you that study methane and sulfur cycling in the deep biosphere, right? We see this is sulfate being consumed as we go deeper in marine sediments, right? And that's happening because there's no oxygen and all the other electron acceptors have been used up. So organisms, bacteria that can do sulfate reduction are consuming the sulfate. And when the sulfate is gone, then the methane can start building up deeper in the sediment. And this is this famous sulfate methane transition zone. And this is where the rates of anaerobic methane oxidation are highest, because they have the two, they're overlapping. They have the methane substrate and still a little bit of sulfate. So that's where those rates are highest. So we can almost kind of, what Kai Hendricks actually did in this 2002 GCube paper is almost think of time, think of depth, in increasing depth in the sediments, almost as, in a way, as going back in deeper and deeper time, which it really is, right? Because sedimentation, your oldest sediment is usually the deepest sediment is usually the oldest. So of course we don't have very many sediments on Earth that go all the way back to even 3.5, let alone much older than that. But we can kind of use this as a framework to think about evolution. And so that's what Kai Hendricks did in this nice paper, thinking that kind of reflecting this figure that probably earliest it was primarily fermentative metabolisms. And once methanogenesis and sulfate reduction evolved, we could start to have anaerobic respiration. Once there was a little bit of oxygen around to stabilize some oxidized nitrogen compounds on Earth, we could start to also use, we could start denitrification. Of course, at the same time, we could also have had metal respiration going on. And then finally, once we had enough oxygen, aerobic respiration could proliferate on the surface of the Earth. And it's important to think about the fraction actually of carbon remineralization that is proceeding by each of these pathways. So this is a figure showing, this is actually depth in the ocean. This is in the water column of the ocean. And this is the percentage of different electron acceptors that are responsible for the carbon remineralization. So essentially in shallow waters, where you tend to be closer to the coast and you have more organic carbon settling through the water column, you tend to deplete oxygen faster, and these alternative electron acceptors can contribute quite significantly actually to carbon remineralization. Once you get to very deep depths where there's less carbon settling through the water column, there's still going to be enough oxygen around essentially to have aerobic respiration basically account for most of the carbon remineralization. So how did life then transition to breathing oxygen? Because obviously after the Great Oxidation Event, oxygen became available, and as I said earlier in the talk, for various reasons, mostly more on the kinetic side than on the thermodynamic side, oxygen is, there's the most efficient proton pumps are associated with oxygen respiration. So this is why I say that about thermodynamics, because actually if you look at the, here again is on the volt scale, more positive and more negative, these different redox pairs, actually N2O nitrous oxide or laughing gas, and also nitric oxide, which is very poisonous for us because it looks a lot like oxygen and it can interfere with our breathing, with our cytochrome C oxidase and poison us. These are more positive. These are so you would expect them actually, these redox pairs, you'd expect just purely based on thermodynamics that they would yield maybe more energy conservation for the cell. And yet that's actually not the case. As I said before, we get the most proton motive force out of the oxygen-water pair. And so this paper, again, does a really nice job. This Chen and Strauss paper does a really nice job of explaining why that is. And there's a few reasons, but as you can see, this is kind of the canonical full denitrification respiratory transport chain. And just glancing at it and remembering back to what we saw for aerobic respiration, you can see there's a lot more components represented here. Now we don't just have complex 1 and complex 3 and essentially complex 4 here. I'll talk more about this, but we also have to deal with these intermediates. There's just more intermediates now in denitrification. And we have to make, or the mostly bacteria that do this process of denitrification, although there are some eukaryotes that do it too, have to make more proteins to basically deal with all of these intermediates and oxidative stress that can come as a result of them. And so partially for that reason, as well as these enzymes apparently just not being quite as good at the proton pumping as the complex 4 is for aerobic respiration with oxygen. Only 6 protons are pumped per electron pair here. Remember, it was 10 for aerobic respiration. So even though it's predicted to be more thermodynamically favorable for these substrates, still we don't have as many protons pumped for this process. And what I really what started me off on this whole thing is this paper I found, which really fascinated me, from 1994, that showed, based just on thinking about the similarity, the structural similarity of these enzymes, showing that basically evolution brought together the second to last and the last enzymes in denitrification to form cytochrome C oxidase. So just walk through this here. So this is nitric oxide reductase. This is the one that looked kind of similar here to cytochrome C oxidase, okay? And this is the copper B site here where nitric oxide is being reduced to nitrous oxide. Here then is nitrous oxide reductase. Excuse me, this is can also be an iron form, I should say. So it gets a little confusing there. I'll have to, we can go into details later. I realize that someone's gonna point that out. Anyway, the nitrous oxide reductase here is also it's copper-based. And the moral of the story is that these two subunits came together, this NOR-B and this part of the nitrous oxide reductase basically came together to create the core of cytochrome C oxidase. And here is the most important, this copper B here, where oxygen gets reduced to water. And they both use cytochrome. Both of these enzymes can use cytochrome C as an electron donor in some forms. There's other forms that use quinol electron donors. But moral of the story is basically denitrification gave rise to aerobic respiration. And along those same lines, it seems that these are called, I should say, heme copper oxidases, because they have heme and copper and they're oxidases. And it's thought that in terms of evolutionary timeline that the nitric oxide reductase form came first. Again, and this is showing kind of roughly age, okay, time going this way. Affinity for oxygen actually is highest for the next form I'll show in a second. It gets lower as we go higher, as we go forward in time, because we have more oxygen around. So the affinity for oxygen doesn't have to be as great for the newer enzymes. And then the proton pumping, as I've been talking about, went from being basically non-existent in NOR, and then I'll show how it's become much more sophisticated as we go forward. So here is one of the structures of nitric oxide reductase. Here then is what's called the C-type cytochrome C oxidase, or CBB3, okay? Here is the B-type, or in Thermus thermophilus, BA3. So you can see now there's more and more residues being kind of added here into this channel for proton pumping. And here then is this canonical version, this classic version. It's also what we humans are using, the A-type cytochrome C oxidase that has this very complex, elaborate proton pumping network that really enables it to get that full 10 protons per electron pair. So this is kind of what's thought to be the case, and yet it's, I think, likely that there are even more types. The more we dig, the more we look through genomes, I don't see any reason why there might not be additional variants that could even be intermediates between these types. So that's kind of the hypothesis that I guess we're in the kind of the long process of testing. It will take many years in order for us to ever characterize something so complex really carefully from an uncultivated organism, but I think that's a great long-term goal. So I know I'm really good at talking for a very long time. So I'm gonna and I know this is only supposed to be 50 minutes max, so I have 7 minutes left before I go over. So I'm gonna have to kind of abbreviate this section. But I'm glad I got through the introduction, because that's really the core, and this is all becomes what we've been kind of thinking about in this context, but we still continue to have a lot of questions about this. So, potential for new respiratory complexes in metagenomes from uncultivated microbes, especially from the deep biosphere. So like I said, we're working on 2 different sites for various reasons that I can talk about in other talks. But we've been working with Sean Crow from University of British Columbia on samples that are from this Archean Ocean analog of Lake Matano, Indonesia. This is a really metal-rich, iron and manganese-rich tropical lake that's stratified, so it has very low oxygen in the surface waters and very high methane in the deep waters. So we were really interested in this starting out because of the methane cycle, but it's led us to all kinds of other interesting findings as well, like this one I'll talk about today. And so Nadia Seinbaum, who is a NASA astrobiology postdoc, actually in my group, she's been working on incubations, long-term, as you can see, hundreds and hundreds of days of incubations of these sediments from Lake Matano on different substrates. And moral of the story is that in our hands, we end up oftentimes with a lot of beta proteobacteria in our anoxic enrichments from these sediments. And often in this group, rhodocyclales. And so Nadia has done genomic sequencing on these, and we've got a nearly complete bin, which appears to be Dichloromonas. It's 84% ANI to other Dichloromonas species, and she has named it Candidatus Dichloromonas occultata for Latin. This is root here is means hidden. And because we've had a hard time cultivating it, we called it occultata for hidden. And what was really interesting is that even though we were actually growing these on methane and manganese, the screening signal from the proteomics that we were doing in collaboration with Brooke Nunn at University of Washington, it actually looked like proteins involved in the nitrogen cycle were actually highly expressed in these cells. And one of which was, one of the most highly expressed actually proteins was a nitrous oxide reductase that uses cytochrome C as its electron donor. Also, we found what was another cluster actually that was highly expressed was something completely uncharacterized that has a bunch of cytochrome-rich predicted proteins. And also, interestingly, at the end of it, what appears to be a heme copper oxidase. We don't know what this is doing, if it's involved, possibly in, we were growing these on manganese, so it's possible that this was involved in the manganese reduction pathway, but it also could be involved in something in oxidized nitrogen or even possibly oxygen, because it has this heme copper oxidase that we're not sure what its substrate is. And this is also interestingly found in other uncultivated organisms, particularly in the deep biosphere. And this is a common theme we see, that looking at these deep biosphere genomes, that often we find things that are totally hypothetical but look really interesting, and they're only in other uncultivated deep biosphere organisms, which maybe makes good sense, but can occasionally make life pretty difficult when trying to figure out what the heck it does or study it experimentally. You will notice, though, that it is that kind of an abbreviated form of it is present in this isolate, leptothrix. So, and this is an iron and manganese oxidizer, and so potentially there's some connection to metals here, but it could be a coincidence. So again, at the end of this operon, we saw that this interesting, what appeared to be a cytochrome C oxidase subunit 1-like protein, and we kind of delved into it deeply, looking at the alignments of it and various other of those heme copper oxidases I was talking about before. And you can see it's quite different from these characterized ones, and yet it shares clearly some of the core residues that are so important for the binding of the hemes, and also of the the copper in terms of this one. It doesn't, though, have some of the other conserved copper binding residues, but you can see these histidines. But then you can see that maybe they've just been shifted slightly down, and it's actually these histidines are being used. So we don't know. It looks really different. You can see there's this huge gap here where this important histidine residue is. This important tyrosine possibly has been moved to here. Anyway, it's all over it's quite different. There's cysteines here. It looks really different. And who knows, maybe it's just some remnant nothing, but it's on a very highly expressed, what appears to be an operon. And we think potentially maybe it's another variant of a heme copper oxidase. We don't know yet, but something for further work. Okay, how are we doing? Okay, it's already 50. So I'm going to have to really crunch through these. So our other favorite site, Hydrate Ridge, offshore Oregon. I've been talking about this for a long time. I promise I'm really going to publish on it. It's embarrassing how long we've had these data and been trying to figure out what they mean. But that's life as a scientist. So I'm going to tell you a little bit more about what I think these might be doing. Maybe. So Ager Bacteria, or Ager Bacteria, some people call them. So these for those of you that study the deep biosphere, you'll see these a lot. These are bacteria that are related to firmicutes, but they're their own phylum. And they dominate in these deep sediments. These, as you can see, this kind of purple color down here, these are the Ager Bacteria or JS1 bacteria. And I think a lot of people have been trying to figure out what the heck they're doing because they're so abundant, but what are they doing? Are they making methane? Doesn't look like it. That would be really exciting, but I don't think that that's what they're doing. So this is just showing that indeed our bin here, this B2 bin, does fall closest to other genomic bins, like from I believe these are oil and gas wells from the North Slope of Alaska, probably also very methane rich. So we found something that looks like complex 1-like in this bin, which means that, interestingly, it's possible that this is capable of respiration, which should be exciting because usually it's only fermentation in a lot of these systems. Fermentation definitely dominates. So whether this organism is capable of possibly doing respiration would be exciting. And we definitely do see what we've called Hun here. It even has what appears to be a regulator for this operon, and it's this really long, I think 16 subunit, I want to say. Yes, 16 subunit, apparently complex, and it looks similar to some that have been studied in, I believe, other hypothermophilic archaea. That this again is a methane hydrate, so it's fairly cold temperatures and it's a bacterium, but it's interesting that it's kind of similar to some hypothermophiles, and it has this more what appears to be kind of similar to those complex ones you saw before, but it even looks like there's a few more subunits that have been added on here, but it has kind of 4 different modules. And in addition to the 3 I talked about earlier, it also has this sodium transporter that's been found in some other organisms as well. And this is just a hypothesis possibly for how, if ferredoxin is the source here, how it could, hydrogen and the ferredoxin could be regenerated. And so a word of caution on protein annotations, of course. I got all excited because I saw anaerobic sulfite reductase, subunit A and B. And I thought, oh, okay, it's a sulfite reductase. But the key sulfite reductase subunit was missing. And it looked like something, in fact, completely different. So these are just similar to the electron transport proteins for sulfite, providing the electrons for sulfite reductase, but that doesn't mean it has anything to do with sulfite necessarily. Okay, so the last breath, in my last breath of this talk, hopefully, just a few take-home messages that I want to leave you with, that thinking about this modularity of respiratory pathways that led eventually to aerobic respiration, and also to be on the hunt for additional ones that I think must be out there to be discovered and characterized, particularly in our beloved uncultivated microbes and candidate phyla microbes from the deep biosphere that could be doing respiration in ways we don't know yet. Here's all the recommended reading. So hopefully this will be recorded and you can screenshot this and take a look at these amazing papers. They'll tell you more than I can ever in this talk. And this is my amazing lab group at Georgia Tech. So I thank them all, especially Nadia, whose work I've really featured here. Thanks very much. And I guess, yeah, Dave, I'll let you decide if I have time for questions or how we want to work this. Thank you.
Thank you very much. Yeah, so I am looking. If I see any questions pop up, I'll let you know. I actually have one for broadcast, and I got my own ignorance. I've never thought about this stuff before, so I have a whole bunch of questions to ask you offline. But
I have a lot of questions too.
in terms of evolution of these pathways, so one of the predictions you might think of, if you have this modularity hypothesis, one of the predictions you might have is that there's more diversity in the earlier components of it, right? So you might expect, I guess not necessarily the sequence level, because they get quenched there in terms of the silent sites, but for the baseline ones, you might expect more variance of those molecules, and then as you kind of work up, you might expect less and less variation in terms of how many different molecules can combine to these to make the cytochrome C in the end. Did you see anything like that in terms of like a bunch of different types of the earlier molecules you might predict, and then less forms of the later ones, or is there anything you can kind of read into the diversity of these molecules to tell you something about how they evolved?
Yeah, I think actually what you just described is actually, I think, kind of what we see is, I would almost, my very kind of rudimentary assumption would actually be that you start very simple and get more and more kind of complex. But you're right that I think we see this toolkit probably having evolved pretty early, and then it seems that some of these pathways, like denitrification, is almost like optimized and stripped down in a way and combined and optimized for aerobic respiration. So they didn't keep all those extra subunits, which I guess kind of makes sense. Whereas I always think of more complex life as just getting extra, kind of accumulating extra things. But I think that that's not the. So you're right. Your way of describing it, I think, is correct, just from the observations I've seen so far.
Cool. There's from what I can see, I'm not seeing any online.
People can tweet at me at Mathanogen if you have more. I'd love to talk more about it.
Yeah, and I'll also say don't worry about the depth of introduction, because like I said, I've never actually, I'm a microbiologist, I guess, now that I've never actually thought about respiratory chains a lot before. So it was actually very good for me to see this, to kind of learn how things are processed and how they actually work. So from the microbial genetic side and pathogens and other things, it's good to see that level of depth. So again, thank you very much.
Thank you for the opportunity, Dave. I really appreciate it.
Awesome.